Okay, everyone. Now I will talk about what I think is one of the most important steps when you do single-cell analysis. Because every time you do single-cell RNA-seq, whether it's MARSEQ2 or 10x, you will have some failed libraries, you'll have some half-dead cells, you'll have doublets. You really need to look at your data, filter your data before you start clustering. Because otherwise, if crap goes in, crap comes out, basically. So I think it's really important to not rush through quality control too fast. And it's often something you have to come back to. So I'll talk a bit first about transcriptional bursting and then go through the different steps of how you do single-cell experiments and talk about the issues at these different points and a bit about filtering of cells. But before we start, I actually want to talk about how transcription happens. Because this is also really important for understanding single-cell data. That in each single cell, a gene is not constantly on most of the time. Expression happens in bursts. So basically, the transcriptional machinery binds to a gene, it starts producing mRNAs. And it circles around that gene locus and produces a lot of mRNA, and then it falls off. So basically, I'll grab a pen, or I can point like this. Here is an illustration from an experiment where you basically see here in black is when the gene is on. You start producing mRNA that is later on degraded. And once the mRNA is produced, you also start producing protein. So also when you measure protein abundances in each single cell, it will vary across time. But the mRNA is much more spiky. So here is basically an experiment where each blue line here is a gene being turned on. So if you have a homogeneous cell population with just one single cell type, and you sample that cell type here, it looks like this gene is really highly expressed. If you sample that cell here, you have no detection of that mRNA. And it's still the same cell type. So it's also really important to keep in mind when you look at the data, not having a gene there or having high expression of a gene doesn't have to mean that much. It's the ensemble of all transcripts together that gives information about the transcriptional landscape. And up here, you can then see sort of also this spikiness in the protein abundance. So that means basically if you have a population of cells from the same cell type, here with only 4 genes, they vary in size. You have stochastic gene expression, so some genes are not there and some are quite abundant in individual cells. When you do bulk RNA-seq, you sort of get the average of this, so you don't really see this burstiness. But when you do single cell, of course, you get some bias that some cells are easier to dissociate and so on. And then you do reverse transcription. And here, you lose also a lot of the transcript, as Jules told you. Normally, we'll detect perhaps 10% of the transcripts or 40%, depending on what technique you're using. And then you amplify it where you might introduce bias. And also, this selection is not completely random. There will be preference for some transcripts to be reverse transcribed more easily than others and so on. So there's a lot of different sources of dropout or missing genes that goes into it, where it's a mix of transcriptional bursting and dropout during reverse transcription that gives all of these zeros in your data frames on the end. So it's just to keep in mind. And then, of course, you end up with these data sets. If you're used to working with bulk RNA-seq, you'll find that single-cell RNA-seq looks quite crappy. But the good thing is that you have a lot of cells instead. So you can overcome a lot of these problems by looking at an ensemble of cells from the same cell type instead. Because we have the amplification bias, we have dropout rates, transcriptional bursting. You may have some background noise signal where you get random priming. And then just due to biology, we'll have bias due to cell cycle or cell state, cell size, and as Jules told, we have a lot of batch effects in single-cell RNA-seq data. It's very seldom I see a data set without batch effects. Oh, it's looking weird now, my slides, but never mind. So the whole pipeline, I tried to visualize you from raw data to gene expression matrix, and then you do quality control before you go into sort of doing normalization, removing batch effects. You try to do clustering or pseudo-time alignment, try to visualize it, and so on. And QC is always the first step once you have your expression matrix. But I seldom find that it's a straight line. You do QC and you get your nice results. You do your clustering, you find something weird with your PCA or with your TSNE, and you see, oh, I have to go back and remove these cells, and you go back to QC again. So it's quite often you go back and forth looking at keep your quality control data in mind all the time when you're analyzing your data. Don't just do QC and think I have a perfect matrix now, because that's hardly ever the case. Oh, my slides are gone. Okay, I should have looked at the PDF. Does it look the same on the PDF on the website? No. It looks okay there. Okay, so it should say all the steps that we have when we go from, so the first step is dissociation, and then single cell capture and lysis. We'll see, now it comes up, good. The red one shows. So this is, I would say, the most critical step in single cell RNA-seq to get good cell dissociation. And it's probably the biggest contribution to batch effects. And it's really important to have whole, healthy cells, short time for dissociation to actually single cell capture. If you have tissues that are hard to dissociate, I mean, you can do laser capture, but you can't get that many cells. It's quite laborious to do. Nuclei sorting is coming a lot. I think it works really well for tissues that you can't get suspension. And I mean, the problems can be you can have multiple cells sticking together. If you have too harsh conditions, you damage the cells. And also, if you damage some cell types and some are fine in dissociation, you will have leakage of RNA from these damaged cell types that will give a background signal. And they will always give a bias to your data. So if you take a bulk RNA-seq of a tissue, and you take the ensemble of all the single cell RNA-seq you did, you will see that the proportions of cells in the single cell and the bulk might not be the same. There will be one cell that you have more of in the single cell because it's easier to work with and it survives better in the dissociated state and during sorting. And there is also a problem that you might get artifacts from the dissociation protocols. So this is one study where they showed actually induction of some genes during dissociation. So they basically stained cells here before and after dissociation. And you can see that there are a lot of genes that are upregulated. And they actually show that you can get artificial clusters of cells that are just dissociation-induced effect. So it's good to keep in mind and check for in your data. And as I said, you might get ambient RNA. This is a data set from Paolo that will come tomorrow, where he's worked with immune cells, where you have 3 different time points. And here are different cell types. And as you can see, all the samples from time point 3 sort of have a background of neutrophil genes, which is also here in the neutrophil cluster. So we think that probably at day 3 we have more neutrophils, but also that a lot of them broke and we have a lot of RNA from that in the soup. And there's not that much work done, as I've seen, for removing background effect. There's one package called SoupX, where you basically take your empty droplets, like Jules showed, and the ones where you're certain you have cells, and you look at what RNA you have in your empty droplets to estimate your background. And then they have some methods to try to remove that from your data frame. I haven't tried it myself, but I think this is something that will come also more tools for fixing these problems. But this will give a big batch effect, of course, if you have background RNA in your data. And then, of course, single cell capture, like we talked about, we can do fact sorting, we can do droplet-based methods, and you will always get doublets, and you will get empty wells. And there might be different, I've heard that there's an upper size limit, but now when we talked about it, some people said they got really big size with DropSeq or 10x. And of course, we might have bias selection of some cell types over others. And the time of sorting is really crucial here to get nice, happy cells. So I think you're all aware of doublets, right? In 10x, there's been some experiments mixing different species and also with cell hashing. So you always get here, we have the human cells and the mouse cells. And then we have some cells that have reads from both species, or mixing of different cell types where you do have some cells in the middle. And the estimates are that it's a linear increase with the number of cells you load and the number of doublets you get. So basically, if you load just 870 cells, you should only have less than 0.5% of your cells will be doublets. But if you're aiming really high, you will get around 8% doublets. And then it's starting to become tricky how to analyze the data. So it's really something you need to think about what you want to do with your data and how much doublets can I afford in the analysis step. And also depending on, I mean, if you have a sample with just 2 cell types, it might be quite easy to find the doublets that are a mixture of these 2 cell types. But the doublets with 2 cells from the same cell type is almost impossible to identify. And that might also influence your analysis depending on what you're planning to do. Another problem is actually that if you have damaged cells, a lot of the doublets you get are actually cell debris from one cell sticking to another cell. This is a data set that I worked with, where I did clustering and then finding cells that have signature from 2 clusters and call them doublets. And as you see, they had a very bad experimental design. We have some samples with few, like around 1,000 cells, and some up here with 17,000 cells. So it's also not advised because normalization and everything will be different for these data sets. Even though we sequence these ones much deeper, we don't really reach saturation here to the same level. But what we also see that the more cells, the more doublets. But then these 2 samples really stood out. And then when I talked to the people in the lab, it turns out they had a problem with in the lab, and they had to refreeze the cell suspension. And then they took it the day after and loaded onto 10x. And basically, I think they just ended up with a lot of cell debris sticking to the other cells. And this one also has lower quality when it comes to all the quality metrics. So there is also one reason to really make sure you have good viable cells to work with, that you get more doublets if you have, or it's not doublets, it's a 1.5 cell, I guess. And when you look at clustering and so on, this is from the cell hashing experiment that Jules talked about, where you have antibody barcodes on your samples and you do it together. And this is predicted doublets with one of these tools to predict doublets. But quite often, you might get a distinct cluster of doublets, and then it's really easy to find them. But quite often you get sort of these streaks in between 2 clusters with cells that sort of have the signature of both clusters. And now you're in sort of the problem. Do you think, like in this case, this group believes that there's differentiation of this cell type to this cell type. Huh? And I think that most of the stuff in the middle is doublets. So if you really want to study the process of differentiation like that, then doublets will be really problematic. because they're going from one state to the other, and you have some cells in between that have the mixed signature, then you really have to find other genes that are coming up in these cells that are differentiating to be able to analyze them. So how do we find doublets? I think this is one of the hardest things. Yes?
Well, I have a question to you.
Yes?
Wouldn't you expect doublets to have a higher RNA count in general?
Yes, you would. But it's, and in SMART-seq2 data, I would say that normally you can see it quite clearly. Not that clearly in 10x from my experience. But it's also problematic when you work with heterogeneous tissue. I actually will talk about this here. Because if you're working with only T cells, for instance, then you should have sort of an even distribution of cell sizes. And the extreme ones are most likely doublets. But let's say you take a tumor tissue, and I remove all the cells with high number of genes, then I actually remove all the tumor cells normally. So then you have to first define the cell types and then look at the bigger ones. But if you have cycling cells, then they will also vary in size depending on where in the cell cycle. If they're just about to divide, then they will be twice as big as a single cell that's not about to divide. So it's not so easy to just look at the number of genes, but yes?
From a perspective, would you be able to tell that the experiments and the doublets just based on what seems together under the microscope?
Yes, I mean, that's something I would always advise. Test your cell suspension conditions. Look at your cells, do a viable stain, count cells, look at them in the microscope, make sure that you know that they are looking fine. Because if you have stuff, cell debris floating around, things sticking together, maybe you need to go back and redo it. But then sometimes people have these rare samples, and they did all their cell suspensions and then they run it anyway, like in this data that I showed. And then you just have to deal with it. But I mean, try to optimize as much as possible to not have these cell debris or doublets. That's my advice.
Or maybe running a certain that lets you keep the cells under suspension for another hour or two?
I don't know. I mean, I think in a way that cell fixing. I mean, there are a few papers out now showing that fixation of cells and running 10x works equally well. I haven't seen that many people using it, but it might be a good solution also, because then you can fix your cell suspension, then you can sort and clean and do whatever, and still have your RNA intact. So I think that might be a good idea if you have a problematic tissue, because some tissues are really hard to dissociate and work with. And if you want to enrich for certain cell types and fact sort, then you will have to have them on ice quite a long time and in the fax machine. So I think that will probably come more to fix cells before doing single cell RNA-seq. Sorry?
Methanol-based?
Yes, that seems to be working quite well. Yes, as I said, high number of genes might work for filtering cells, but it's problematic if you have more complex tissues. And after you cluster, look at sort of your T-SNPs, your clusters. And maybe if you have a streak in between that is also high in number of detected genes, that could indicate that you have doublets. And also, you could sort of have a feeling for the doublet rate if you know how many cells you loaded on 10x, if you know you don't have cell debris. So you can sort of try to estimate how many doublets you see. But then also you should keep in mind that, let's say I have a sample with 90% T cells and a few other cell types, then most of my doublets will be to T cells, and those I will never see. So then my doublet rate will look lower than what it actually is. And there are some tools. There's a lot of publications coming up just now about different tools for predicting doublets. Most of them are based on the similar idea, is that you take your data set and you simulate doublets. So basically you take your single cells and you merge 2 cells and call it a doublet. And then you do some clustering or nearest neighbor analysis. They're using different machine learning techniques to do this. And sort of find all the cells that are close to the simulated doublets you predict with a high likelihood to be doublets. And then you can filter based on that. And I find that it works quite well on heterogeneous tissues. But when you start having, like I said, 90% T cells, for instance, most of my simulated doublets will also be T cells. So I've only run this doublet finder. And then there's some tools, like in scRNA, there's a tool for detecting clusters that are doublets that have the signature of 2 different cell types and so on. So I think some you have to do your analysis, do your t-SNEs and clustering, go back and look at doublet predictions. I always end up going back and forth a bit until I really decide what I believe to be doublets. And fact is, I think when you have a lot of doublets, it's impossible to get rid of everything. Yes? Yes, that's very tricky when you're looking into cellular differentiation. Hopefully, during differentiation, if you go from one state to one state, you will have some genes coming up here in the middle, right, during differentiation time, so that you can distinguish what's being differentiation to just doublets being a mix of these 2 signatures. But if that signal is weak and you don't really know what it is, what you're looking for, then it can be hard. So try to plan to have less doublets if you're looking into differentiation is my advice. Yes?
How these filters then work with the doublets that they found? Can you use this information or you just filter them out?
I mean, you can either, normally I would just get the scores for like doublet prediction. I would do my clustering and everything. I would basically plot it like this and then look, oh yeah, this really looks like doublet. This looks like the correct prediction. And then I would decide what to do with it.
So you use just the score to figure out how many of the doublets can you see here?
Yeah. And to sort of decide if I believe them to be doublets or not. It is very hard. It's not like you can get an answer right away. If you have 2 really distinct cell types that are not supposed to interact, then it's easy. But it's, with all the interesting biological questions, you often have these kind of problems that you have something in between and you don't know. So it might actually be that you have to really dig into what the genes of this cluster is. Do you find something that's interesting, or do I think it's doublet before you remove it? It's never an easy case. More questions on doublets while we're at it? Nope. Let's see. And then, once we sorted them, we need to lyse the cells. Now, when you run 10x, it's all a kit that you run, and you don't really know what's happening in the background. But it actually is a difference for different cell types. They might require different lysis conditions. If you're working with SmartSeq2, doing it on your own in plates, you can optimize this. I know people are trying to do it on plants, for instance, where you have to break cell walls, and then lysis conditions versus what damages RNA and what interferes with the library prep is important. What I've seen also in data is that depending on the cell type, you may or may not get nuclear lysis of your cells, and that will give a clearly different transcriptional landscape, because the nuclear transcripts will be detected or not depending on if you have nuclear lysis. So that can also be important to think about. And then reverse transcription, of course. This is the big limiting step. If you detect transcripts or not. Hopefully, it will be fairly, I mean, it's not a random selection, but hopefully it will be fairly similar for all cell types, that the same transcript will have the same chance of reverse transcription, that will hopefully be able to use the data. And then, of course, we amplify. If we have UMIs, we can control for it. But there will always be a bias there. And as showed you these fast QC plots, we can also have issues. People tend to forget this. You think the sequencing machine is always working perfectly, but it's not. You might have just an air bubble or some contamination in your sample or something that will make part of your flow cell fail, and you will have a lot of low quality reads. But I would say that you need to, when you look at fast QC reports for single cell data, you will often say failed, 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 failed on all the steps, just because of the nature of single cell data. You have more primer dimers in single cell data. You have a lot of bias in the beginning due to all of these primers that we have and so on. So, but it says failed in the fast QC report, doesn't mean that it's completely crappy, but at least have a look at it. And especially if you see parts of your flow cell failing, it might be a good idea to actually resequence your data. And also, depending on what machine you have, you might have problems with index swapping, where you get reads from one cell with the index of another cell, and then you're really in trouble. There are some tools to try to fix this out there that you can look into. Okay, that was the steps of the library prep. I will talk briefly, since everyone here is using 10x. This is not relevant to you guys because we can't use spike-ins in 10x. Because as Jules said, most of the droplets are empty. And if you throw in spike-in into everything, then we'll mainly just sequence empty droplets with spike-in. And that will cost a lot to sequence before we actually manage to find the cells. So there are two kits used with artificial sequences that we add to the cells. And you should add it in the lysis buffer so that they're sort of going through all the same steps as the cell RNA as they're being processed. And basically the idea is that if you have these spike-ins, because these two cells will look like they have an identical transcriptional landscape, but if you add in spike-ins, you can actually see that you have twice as much RNA in this one compared to this one. It's just the same abundances of the RNA, or relative abundances. So it's not easy to make a perfect control and a perfect spike-in. And we do have a difference in amplification bias because these ERC spike-ins are shorter, transcripts amplify better. But we can still sort of estimate back the number of molecules of RNA using the spike-in. We can use it to model technical noise, dropout rates. So basically for each experiment, we can see how efficient the reverse transcription was in that single well or whatever you're using. And sometimes in the beginning, people try to use it for data normalization, but it turns out that normalization by spike-in is just introducing noise almost. Yeah, that was it. So now quality control and filtering cells. I put on a long list of things that people look at. If you look in literature, in different papers, they use different kind of quality control filterings. And it's really up to you to decide what you want to use. I'll go through some of these. Just looking at sort of the mapping. Now when you run Cell Ranger, it's all hidden. You don't get any mapping reports per cell. You only get per sample. So you can't really get to that. If you use other pipelines, you can actually get per barcode how well the mapping went for each barcode. Because in some samples, you will have more primer dimers just due to low amount of RNA or other problems in the library prep. And so the number of reads you have, or sort of the percent uniquely mapping, how much maps to exon or to mRNAs, and so on, can actually tell you a bit about the quality of the cells. So when it comes to SmartSeq2 data, I always look at these stats. But when I have the cell ranger report, I don't, because I can't find it. And basically, the cutoff number of reads might be that if you have too few reads, there's not enough information in there to actually do something about your data. When it comes to spike-in, it's detection of spike-in. If you don't even amplify the spike-in and detect it in a library prep, you know it's crap, that you can't use it. So then you can throw away that library. And the relative proportion of spike-in to RNA can tell you something about how much RNA you had in the cell, and you can use that also for filtering. Yes? Yes, this is really tricky, and it depends clearly on what cells you're working on. Because if you're working on big cells, then you can add more spike-in. If you're working with very small cells, you might end up in a situation where like 10, 20% of all the reads you're sequencing is spike-in, and then it's sort of a waste of money, I would say. So it is not so easy. So I think it's a good idea to look at what other people had for different cell types. Should be around a couple of percent spike-in, I would say. Yes?
Yeah, so, in order to do so, you either use spike-ins for humans, or you can use them both in the same sample?
I mean, techniques where you use UMIs, you can use spike-ins, yes, but not in DropSeq. But you can do like the STAR2I method, where you're doing microwell systems. You can actually also use spike-ins, and it's also UMI based. But once you're starting using these microfluidics droplets, then you'll have every droplet with spike-ins, also the ones without cells, so then you can't use spike-ins.
Mm. Okay, so with the droplet you use UMIs? No.
Yes.
Okay. But if you don't have either UMIs or spike-ins, can you still do the read down?
Yeah. I mean, you can. I think the spike-in, to me, the most important is for the quality control to have spike-ins that I can see sort of if the spike-in libraries were amplified or not, and so on. And also for looking at technical variability, also batch effects between samples and so on. Then it can be good to have the spike-ins, but it's not lost because you didn't have spike-ins. Mm-hmm. You can still work with your data. Yes. Okay. One thing to look at, number of detected genes, as we already talked about a bit when it comes to doublets. It will give you an indication of if it's a single cell. I mean, in this very example, actually, they did some samples with 100 cells, with single cells. And then we have clearly some failed libraries out here. So when you have this really nice homogeneous population with the same cell type that should be the same size, then I'm fairly confident to just use number of detected genes to filter out cells. But if I have mixed sizes of my cells, it's harder. And it's also something to keep in mind. This is actually innate with cells. If you look at spike ratio here versus forward scattering, there should also be a rough estimate of the size of the cells. You see that it clearly correlates, that bigger cells have a bigger ratio of RNA to spike-in, and they also have higher number of detected genes. So it all follows together, all of these measures. So you might not have to look at all of them since they also correlate. But you have to keep in mind that you don't want to remove all the big cells in here if there is a variation within your population. Mitochondrial read fraction is also something that most people use for filtering. And it's not really been shown. There's been one experiment where they actually put the cells to stress and showed that they got an increased mitochondrial read fraction. The theory is that if the plasma membrane breaks, you might get leakage of the RNA from the cell, but the mitochondria are still there. So you get a higher proportion of mitochondrial reads. It can also be an indication of apoptosis, that the mitochondria become more active if you have mitochondrial apoptosis. So normally, high mitochondrial content might indicate that you have some problems with these cells, but sometimes you have a mixed issue. If you're doing fat cells, for instance, you have higher mitochondrial contents than some other cell types. So it's still knowing your data and what to expect from your data when you're doing filtering is really important. And as Jules talked about, ribosomal protein fraction, I've also seen in scRNA-seq data that you actually do get some templating of ribosomal RNA if you have a lot of RNA fragments. I've seen actually that samples that's been in the freeze for a longer time tends to get higher RNA fraction, especially for some other species than human and mouse that I've been working on. So this also clearly relates to the quality of the cell. But when it comes to ribosomal protein fraction, it's also the same thing. Depending on the cell type, you will have different ribosomal subunits, and the different ribosomal subunits can be really important to define a cell type. So in some cases, it will vary quite a lot, and you will have significant differentially expressed ribosomal proteins that are really important for biology. And in other cases, like Jules showed this morning, it's only related to quality and the stress of the cells. So it's also, once again, something you have to think about biology of your system. But having ribosomal RNA is for sure a sign of RNA degradation, I think. When it comes to scRNA-seq, we can also look at 3' bias, because since we capture the genes at the 3' end, if the RNA is starting to be degraded, you will only get the 3' end of the genes, and you will get a gene body profile. So here's 3' to 5' end coverage. And if I have few cells that look like this, I would throw them out. But if they look like this, I would be happy. But then you might have a case where all cells look like this, and this is the data you have to work with, and then you'll have to just work with it and realize that you catch what you can. So back to this long list. I would say these ones and this that I highlighted here in bold are probably the ones that I think are most important to look at, especially when it comes to scRNA-seq, then these two also, and in 10x is genes and mitochondrial read fraction. So then how do you filter? I say look at the distributions. Look at how it looks for each sample you're doing, because then you get a feeling for sort of out here, it's all crappy. But then you might have, for instance, red blood cells that are really tiny with low amounts of RNA that are out here. And then it's up to you if you want to keep the red blood cells or not, because depending on what your question is. Here is sort of how many reads we have at the 3' end, where it's extremely high for some cells, then I would take them out. Gene detection here, I would say these are clearly doublets here and failed libraries. So I always look at the distributions and try to make some reasonable cutoffs for each sample. You can also actually just take all your different quality control metrics that you have and throw them into one big matrix and do PCA and find outliers in the PCA. That's actually quite a good way to find the outliers, but it might also be that the outliers you get in the PCA are your small cluster of tumor cells, for instance, that are much bigger and have much nicer QC metrics. You have another outlier of red blood cells or something that you want to keep in. So So look at what you get before you remove anything. And in drop-seq data or 10x data, something that a lot of people are using, on one part just looking at number of molecules, sort of cutting at both ends, but actually plotting number of molecules versus number of genes. And if you have a lot of points down here in this region, there are a lot of UMIs, but still very few genes. These are commonly removed also. So there are, as I said, not one easy way to filter. You have to know your data. You have to know how heterogeneous, what kind of cell types, cell sizes you expect from your data set, and look at that when you apply filtering. And I would always calculate my cases that the clearly fail libraries where I only detect like 50 genes or have very few reads, I filter right away. But then I do some PCA, I do some t-SNE, look at QC stats. Then I go back and I filter what makes sense to me. And then I go and do new clustering and t-SNE and PCA. Because it is a bit going back and forth. Another thing is quality control on genes. I think during the afternoon we'll talk about a lot of gene set selection, finding variable genes, and so on. But there might be some problematic genes in your data set. So here is actually a plot with a contribution to all total counts per gene. And as you see here, this is clearly a case with a bit too much spike-in. These are the spike-in genes. There are a big proportion of all the reads. And then there's a gene here that actually some cells have like, there should be some lines up here, like 25% of all the reads is this one gene. And quite often that one gene is MALAT1. It's a nuclear link RNA. There's other nuclear link RNAs that also pop up here. I don't really know why, but it seems to clearly correlate with quality of the cells. And that could be also plasma membrane breaking, all the cytoplasmic RNA leaking out, and what you're left with is nuclear and mitochondrial RNAs. I can show you. So here are some plots from a data set I have. Here is mitochondrial proportion and MALAT1 proportion. So there is a clear trend that it correlates. So look at these kind of plots, looking at what genes I have in the data, I make a decision on what genes to keep. Another thing might be that you have some genes that make sense to have in there, but they're 20% of all your reads. And it varies some between, you might have actin, hemoglobin, depending on what sort of cell type you're working on, that will skew the normalization of your data. Did you have a question? MALAT1 pops up in both SmartSeq2 and 10x that I know. And in both mouse and human. And that's as far as I know. But there are other link RNAs that often come high also together with MALAT1. I don't know all the names of them. But it's good to keep in mind. And also, I mean, if you have these genes that are really highly expressed and are supposed to be there in all your cell types, it could be a good idea to remove them just that because they will skew the whole normalization of your data set. If they are 20% of all the reads in one and they're 40% in one, all the other genes will be scaled depending on just that one gene. So if you have some extreme genes up here, it is good to remove them before doing clustering, so not to keep them in the clustering step. And like we talked about, these ribosomal genes might be a good idea to remove, especially if you expect to have one cell type in there. So you need to filter cells and you need to filter genes. before you start. And when it comes to mitochondrial genes, I never keep them in when I do clustering. I keep them in for mapping for QC reasons, and then I take them out before I take the data frame to clustering. Because I think in most cases it's just technical bias due to quality of cells. And Jules talked about this quite a lot. As we said, batch effects will happen. And I think it's quite important to take all your quality control metrics and plot them per batch. So that you can sort of look at uniquely mapped in your different samples or detection of spiking in your different samples and so on, so that you can understand also what your batch effects are. Just knowing that you have a batch effect is quite important, but I think it's also good to understand why you got a batch effect, and then maybe next time you do an experiment, you will be able to do it better. And I think PCA is really important for QC. I use it a lot because PCA gives you really good understanding of your data. What's the main variability in your data? And this is something that I always see when you do a PCA on your data set. The one or two will correlate almost perfectly with number of detected genes. And of course, this might be biological as well, that you have smaller to bigger cells. But in many cases, it will be technical, just due to different dropout rates in different library preps. And then there are methods to regress out this before doing analysis, or try to fix it with different types of normalizations. You can also use PCA to check for batch effects. We'll look at this a little bit during the exercise, if you have time to reach to that step. So basically, you can look at your different batches and how well they correlate to different principal components to start understanding how much batch effects you have and what they are, because then you can also go into the loadings of PC1, for instance, here, and see what are the genes that are driving the separation of these two data sets, and really understand what you have in there. And you can actually sometimes use it to detect contaminants. Here is one project where we're working with PDX3 EGFP cells. And then we have clearly in the PCA these cells that we didn't really want in the analysis. So just based on PCA, we can remove all the cells that we don't want to analyze. So that's it. Almost on time. Try to plan so that you don't have to do so much QC. If it's all very consistent, very good cell, even number of cells with not too many doublets, then QC will be fairly easy. But it's quite often the case that you have some failed libraries anyway. And really think about, what data do I have? What kind of QC stats should I expect from this tissue, from this cell type? How much variation in size is there, and so on? And as I said, finding doublets is really hard, so try to avoid overloading of cells. And as I said, when you've done clustering and you think you have really nice clusters, take all your QC stats and plot them per cluster. And then maybe you see like you have one cluster with high number of detected genes, one with low number of detected genes. Does this make sense biologically? Or is it just an artifact of technical bias? So I always, in the end also, when I've done all my analysis, I take my QC stats again and look at that in the data. Because I think that really helps to understand what the clustering is. Because Jules showed you that you can get clustering of the cell population just by random. And you can for sure get clustering due to number of detected genes, or how much mitochondria or MALAT1 you have in your sample or something. So you will get technical bias in your data due to library quality that might drive clustering.